So I think without further ado, we can go ahead and get started with Robert Huff, and you can go ahead and share your screen with us and get us started.
Sure. All right, let's see. All right, can you see just the regular PowerPoint slide right now?
Yeah, it's right there.
All right, cool. Thank you for having us today, Dr. Kearney. So my name is Robert Huff, and I work in the lab of Dr. Jason Pitts. I'm a PhD candidate, and today I'm going to be talking to you about characterization of vector mosquito olfactory receptors. So let me just go ahead and hide these faces. So there we go. All right, mosquitoes cause dramatic morbidity and mortality throughout the world. They're one of the deadliest animals. And the reason for this is because they carry a number of different disease pathogens. So these can be viruses, protozoa, or parasites. And another thing about mosquitoes are they're highly invasive. So due to climate change, their habitation ranges are dramatically increased. So what I'm showing you here is, let me see if I can get a little pointer real quick. There we go. Can you see that pointer? I'm going to take that as a yes. So what we're seeing here is the distribution of Aedes aegypti mosquitoes throughout the world. So the hotter the color, the more months throughout the year in which they're able to transmit disease. So, oh my goodness, sorry, this isn't letting me. Let me just get rid of that. There we go. So what this group has done is they're modeling how distribution is going to change in relation to how the climate changes throughout the world. So you can see here, especially in the northern latitudes, we're going to get a dramatic increase in the population ranges of these mosquitoes, which is a bad thing because that means there's going to be more vector disease prevalent throughout the world as time goes on. And another thing about mosquitoes is they're becoming more and more durable. So they have acquired what is known as insecticide resistance. So insecticides are chemicals used to treat areas that are infested with mosquitoes to try to knock down their population sizes in the hopes to not only decrease population, but also decrease the amount of disease that is transmitted. And this occurs through a number of mechanisms shown on the screen here. And that is not a good thing because not only are mosquitoes becoming more and more resistant, but so are insects in general. So not a good thing at all. So I want to start first. I just realized that didn't change, sorry. I want to start first with just the mosquito life cycle, just so you have a good understanding of how mosquitoes work in the world in case you don't research them on a daily basis like we do. So I'm going to start with oviposition site selection, which is on the bottom right there. So a female mosquito will lay eggs in or on or near water. The eggs will undergo development for a couple of days where they will hatch into an aquatic larva form. So these larvae live in the water and they need to find a food source that can be bacterial or fungal cells, as well as dead or organic decaying matter. So the larva will transition into a pupal stage where it will undergo complete metamorphosis and emerge as a flying adult insect. So the first thing that mosquitoes typically do, they'll need to go and find a plant host to take a nectar meal. So both male and females require nectar as their sole carbohydrate source. Typically what happens after that is they'll kind of fly around in the environment for a few days and kind of mature a little bit, and then they'll end up mating with one another. And the next thing that needs to happen in the case of females is they need to find an animal host, whether that's a human or some other animal, to take a blood meal from. The blood is required because it's very high in protein that is necessary in order for them to generate a batch of eggs. So once they take a blood meal, the females will need to rest and digest that blood meal and actually generate the eggs. This typically takes about 3 days. And then at which time they're actually able to go find a new oviposition site so that they could lay their eggs once again, and the whole life cycle starts over again. And this happens pretty rapidly in the environment anywhere from 2 to 3 weeks with good temperature and humidity levels. So I wanted to describe the life cycle to you because anything you see on this figure that is in red has a very important aspect of chemosensory biology that's connected with it. So in order to find somewhere to lay eggs, to take a blood meal, to find nectar for carbohydrate resource acquisition, all of these behavioral traits require the ability of a mosquito to detect volatile odors in the environment, and they do this with their complex chemosensory system. So I'll go into a little bit more detail about what that system looks like. So we have an adult mosquito on top and a larval mosquito on the bottom, and they have a number of different sensory appendages, all of which are decorated by small hair-like protrusions known as sensilla. These sensilla are going to be perforated, so they have pores that allow odors that are found in the environment to enter the sensillar lymph, which is a fluid-filled area that has dendrites of olfactory sensory neurons present. And if we take a closer look at the membrane of the neuron, we can actually see that they are embedded with small receptor proteins. In this case, we're looking at chemosensory receptors. So the odor can travel from the air outside in the environment into the sensillar lymph and bind to a sensory receptor. When this happens, an action potential is generated that will ultimately lead to a signal that travels to the insect brain and leads to some sort of behavioral change. And the receptors that we study in the lab mainly are from 3 different classes. We have ORs, or odorant receptors, IRs, or ionotropic receptors, or GRs, or gustatory receptors. And off to the right there, we have some compounds that they're typically associated with detecting. So when the molecular interaction of a ligand and a receptor occurs in the insect's sensory appendage, this will lead to a number of different behavioral changes. A lot of these mentioned just in the previous slide. And when we can better understand how these molecular events lead to behavior, we can come up with ways to possibly interfere or change these behaviors. And this could influence the amount of disease spread that occurs due to mosquitoes in the environment. So in the lab, we study this in an interesting way. We utilize frog eggs from the frog Xenopus laevaeus, and we use these eggs in order to express our olfactory receptors of interest. And the way we do this is through microinjection. So we'll generate messenger RNA transcripts of our receptor of interest, and we actually inject the frog eggs. These transcripts will be translated into functional membrane proteins, and they'll act just the same way they do in a sensory neuron of a living mosquito. So the way we can measure and record from these oocytes is using a technique called 2-electrode voltage clamping, where we use 2 small microelectrodes that pierce an oocyte that is expressing our receptor of interest. And then what we can do is we could take a compound library of many different chemicals and actually perfuse these odors across the oocytes that are expressing the receptors. If one of these compounds from our library binds to the receptor complex we're studying, we can actually measure changes in current due to these ion channels opening and closing, and that looks a little something like this. So we'll have a baseline measurement way up at the top, and then we'll give it an odor stimulation. And if it responds, you'll see one of these dramatic drops in current that will ultimately come back to baseline, and we could stimulate it again with a different compound. So what we can do is we could take this digital information and turn it into easily readable graphs, something that looks like this, so that we get a good idea of what these different receptors are responsible for detecting in the environment. So the first little bit of data that I'm going to be presenting today came from a publication that we put out in 2019 titled An Odorant Receptor from Anopheles gambia, that demonstrates enantioselectivity to the plant volatile linalool. I know that's a mouthful, so I'll go ahead and break down the title for you. So Anopheles gambia is the malaria mosquito that is primarily found in Africa. And linalool is a plant volatile that is associated with flowering plants. And enantioselective means that this compound exists in one of two forms due to a chiral carbon that creates two different stereoisomers. And lastly, the odorant receptor that we're studying is OR29 that is highly expressed in the labella of both male and female mosquitoes. So the labella is at the tip of the proboscis, or the mouth part that mosquitoes utilize for both blood feeding and nectar feeding. So I'm showing you here a phylogenetic tree of two different odorant receptors. On the bottom, we have OR29, which I just went over from a number of different Anopheles mosquitoes. So all of these receptors are anywhere from 75 to 98% identical, so they're highly homologous. And on the top half of the graph, we have OR53, which is anywhere from 65 to 94% identical. And if we were to just compare any two of these receptors to a different receptor, they are all anywhere from 58 to 98% identical amongst each other. So these are two very homologous, well-conserved receptors across a number of different species of mosquitoes. So they probably have a very important implication in the chemical ecology of these mosquitoes. So the data I'm going to show you going forward is going to consist of two OR29 homologs from Anopheles stevensii and Anopheles gambia, as well as two OR53 homologs from these same two species. So just to give you an implication of how important these mosquitoes are, I'm showing you the distribution of different malarial vectors throughout the world. So in red in Africa is primarily where Anopheles gambia is going to transmit malaria. And in pink in the Indian region is going to be Anopheles stevensii. Although a lab here at Baylor, Dr. Tamar Carter's group has shown recently that Anopheles stevensii is starting to migrate into the Horn of Africa region and start to compete with other vectors that transmit malaria. So we'll go ahead and get into the data now. So using the oocyte system, I express our receptors of interest, and then we go ahead and test them with different chemical classes. So it's pretty obvious that alcohol blend number 3 here gave robust responses to all 4 receptors. So what I did next was I went ahead and took the 9 compounds that comprised this blend, and I tested them all individually in the oocyte system. And I came up with this data. So you could see S plus linalool here and R minus linalool here. We're giving pretty good responses to all 4 receptors. And interestingly, if we take a look at OR53, which is in blue from the 2 species, we actually almost get comparable response profiles from the 2 enantiomers, which is interesting, and I'll go into detail here in a second. So I was able to test these 2 enantiomers at many different concentrations in the same oocytes and generate concentration response profiles. So what this allows us to do is calculate EC50 values, which is shown there in the legend. And what an EC50 value is essentially how much of this chemical is required in order to get a 50% response. So the less amount of an odor that you need to get half of the maximal response means that it's going to be the more effective ligand. So in this case for Gambia OR29, the S plus linalool in blue was giving really good responses, about a 2-fold reduction in EC50 value. And we see the same thing when we looked at Anopheles stevensii OR29, about a 2-fold reduction. But when I went and I tested out the OR53 homologs, I actually saw the complete opposite. So in this case, the black line for R minus linalool is going to be on the left-hand side of the graph, signifying it's likely the better ligand for this given receptor. And the same thing held true when I took a look at Anopheles stevensii OR53. So to summarize this information for you, basically what this data is indicating to us is that the OR29 genes are likely going to be responsible for detecting S+ linalool in the environment, and the OR53 genes are more likely going to be detecting R- linalool in the environment. So that is some data that we're actually just about ready to submit to a journal in the coming weeks. So pretty excited about that. But I wanted to go ahead and kind of give you an idea about why this is important. So a reminder that linalool is a plant-based volatile, and it is associated with flowering plants. Here's an image of 2 different Anopheles mosquitoes feeding on plants. So the idea is that since this compound is associated with plants, it might be an indication to nectar-seeking mosquitoes that they might have a suitable plant host in the given environment that they can go take a nectar meal from. And another reason this is important is because we could utilize this information in the form of attractive toxic sugar baits. So a toxic sugar bait is essentially a bait and kill station that is composed of a sugar solution, some sort of attractive odor, and a toxin. So when a mosquito comes and finds this, it'll take a sugar meal, but shortly after doing so, it'll go and fly away and oftentimes either die or not be very reproductively successful due to the toxin itself. So this is a way that you can actually control mosquito populations in the local environment. And we can use the information that we get from experiments like the oocyte expression system to try to develop novel odor ligands that are both activators and inhibitors of different mosquito behaviors, so that we could use these in combination in something called push-pull vector control. So if we're able to mask or push mosquitoes away from humans or other animals and pull them into traps so that they die or are unable to transmit disease anymore, we can decrease the likelihood that different pathogens are going to be introduced into the human population. So that's just an idea of how we could utilize the information that we get from these experiments and make them have some sort of real world application that we could use going forward. And just real quick, I wanted to quickly state that we actually identified an OR from a different species of mosquito that seems to be doing the same sort of detection of linalool-like compounds in the environment. So this is something we're going to follow up, and hopefully we see something similar to what we do with Anopheles species of mosquitoes. So now, quickly, for the sake of time, I'm not going to go into too much detail, but this is a figure from a grant that we had funded this past year that looks at a number of different ORs from 4 different species of mosquitoes that we think are likely going to be responsible for detecting different plant volatile odors. So they have some sort of behavioral impact, whether that's in nectar seeking of mosquitoes or oviposition site selection. So I'm going to focus in on 3 receptors in this small clade here that I've identified to detect a compound known as camphor. So you can see in pink there on the far left-hand side of the graph that this receptor from Aedes aegypti OR49 detects this compound and generates really good concentration response curves. And the same thing holds true for OR49 from Aedes albopictus, which is another mosquito, the Asian tiger mosquito, that detects the same compound. And lastly, the same thing we found to be true for Culex quincefasciitis OR38, which is a homologue of OR49 that is detecting this same compound. So why is this important? Well, camphor, like you might have guessed, is a plant volatile, and it is actually already known to be repellent to a lot of different mosquitoes. So we might have, discovered the molecular interaction between an olfactory receptor and a compound that already has the known behavioral output, which is really cool. It's always good to be able to say you discovered something. And due to the sake of time, I'm not going to go into any more detail, but we've also made a lot of progress on this big chart here, and we've actually been able to identify ligand receptor interactions for about a dozen or more of these receptors so far, and this is ongoing work continuing to go forward. And lastly, for a second project utilizing a different type of receptor in IR, we have the same type of research ongoing where we're trying to determine what this clade of receptors responds to. In this case, we think it's going to be carboxylic acids, which are components of human odors. And we've actually been pretty successful so far, and we've been able to identify this clade of receptors here that responds to a compound nonanoic acid, which is shown in this figure here up above, to be a component of human odor. So this compound might be an indication to mosquitoes that a blood meal host is nearby to take a blood meal from. And other than that, I think that's just about all I have to say. And for the sake of time, I'll go ahead and thank everyone in the lab for everyone's help and support throughout the research. And I'd like to thank our collaborators, Jonathan Bobot, Matt DeGennaro, Omar Akbari, and Jeff Riffle. And of course, I'd like to thank you for coming and listening. And with that, I'd be happy to answer any questions.